Thanks very much for that introduction, July, and thank you all very much for coming this morning, both virtually and in person. So today I'm going to be talking to you, as the first slide says, about a new dimension in marine biodiversity. But first, to sort of set the scene, one of the questions that I'm particularly interested in is why species are found where they are. And this is a question that naturalists often ask implicitly. Based on my past experiences of where I've seen species, I can then generalize to infer where else I might find more. For example, I learned to fish from my grandfather. He showed me where to find the fish in this particular lake. And then based on that experience, I was able to generalize as to where to find more fish. I should also mention that every time I give this talk, that fish gets a little bigger. It's interesting how that works. But this works well at a local scale because we have an experience of these things and we have sort of a qualitative model in our heads about where species are found. But this tends not to work very well at a global scale. You can ask a fisherman where to find fish in the system that he's familiar with, but ask him to generalize worldwide on where to find fish, and he'll be a little bit more challenged. And so this is why I got into sort of big data in order to answer these questions in a more quantitative and repeatable framework. And to kind of give you an idea of my mindset as far as how to find species, where species are found, just as sort of a general example, I tend to do a lot of modeling of distributions. So in this example, I've got this species of netted pawpaw, Asimia reticulata, which is native to peninsular Florida. And each of the black points is a presence. Each of the hollow squares is an absence. And I can then use this data to model where the species might be found, or where additional locations of the species might be. For a variety of applications. So I have my occurrence points. Based on those occurrence points, I might have environmental measurements that I can then plot. In this case, we have temperature on the X and precipitation on the Y. All of these black dots correspond to occurrences of the pawpaw. Based on that information, I can then come up with some sort of mathematical model of where the species is found, which in this case, I'm calling an ecological niche. There are a lot of different ways that ecological niche can be used. This is how I use it. And then based on this information, I can then project that niche back into geography to estimate where the suitable habitat for the species is found. And so I use this to approximate the distribution of the species. And this is sort of the basic building block of a lot of the macroecological work that I do. And this approach can be used in a lot of different applications, things like targeted sampling for rare species. So in the case of that pawpaw, I might be looking for additional locations for conservation. I could also use it for invasive potential of introduced species, changes in suitable habitat with climate, designing protected areas, or biogeography. And it's this last application that is mostly what I'll be talking about today, and where a lot of my work is currently focused. So the big question that I'm asking is what drives patterns in marine fish biodiversity? And this is a very fundamental question for a lot of macroecologists. Not marine fish necessarily, although I would argue that it's more important than many people tend to think. So this question is interesting because while there's been a lot of work on this topic in terrestrial systems, marine systems have been comparatively neglected. So you might have a lot of work that's been done in terrestrial systems with regard to the latitudinal diversity gradient, for example, is a really common one. But we just don't have the same focus in marine systems. But the predictions are often very similar. So for example, we might expect more species to be found the closer you get to the equator, this latitudinal diversity gradient that I mentioned. And there are a lot of explanations for why this might be. Things like energy, primary productivity, those things there tends to be more energy and more insulation the closer you get to the equator. Age can be a factor that's often used to explain the latitudinal diversity gradient, that habitats that are closer to the equator tend to be the kinds of habitats that have existed for longer in the Earth's history, and also just that there are generally favorable conditions. And all of these things are used as explanations to say, well, all of these species pile up near the equator, and then we get much lower diversity toward the poles. And this is actually a study from Rybowski et al. In 2018 on marine fish. So they have latitude on the X and then species richness on the Y. And so each of these dots represents a cell across the whole world. So while there's a lot of variation in the amount of diversity, in general you get higher diversity near the equator, and then it drops off as you get closer and closer to the poles. Another explanation and another hypothesis for why diversity is found where it's found that's often invoked is the nutrient gradient. That there tend to be higher nutrients the closer you get to the equator, and that corresponds with high productivity. And that high productivity can also promote diversity because there are more resources available for more species. So in this example, this is another marine example from Chowdhury et al. In 2016. So at the top, this is primary productivity in the ocean, and then we have their map of species diversity. So areas with high productivity tend to correspond to areas with high species diversity, although you might see that there's a little bit of a mismatch here. The problem with the approach that these studies have taken is that they're really only scratching the surface, and in this case, I mean literally. So this scatter plot shows apparent oxygen utilization, which is one of the environmental variables that we often use in marine systems, and temperature on the Y. And each of these points represents a global, a representation of the combination of these variables in the Atlantic. I'm focusing today on the Atlantic for computational reasons. So each of these dots is a point on the surface, but what we see when we look at the full picture is that there are a whole lot of environmental combinations that aren't represented when we talk about the surface. And those two example studies, the Rybowski study and the Chowdhury study that I presented, those are both focused on two-dimensional distributions of these marine organisms. So they're only being mapped in 2 dimensions. They're using the same distributional model approach that I introduced with the pawpaw, using the combination of environmental variables at latitude and longitude, but they're neglecting depth. So,
what is apparent oxygen utilization?
So apparent oxygen utilization, I had a question in the audience about what apparent oxygen utilization is. It's basically, it's a comparison of how much oxygen is in a system and how much oxygen is being used. So at the surface, there's a lot of oxygen that's dissolved in the ocean, whereas at depth, you tend to have very little dissolved oxygen, but the reason that there's little dissolved oxygen is a combination of the dissolved oxygen, but also the amount of oxygen that's being used by the system. So the more oxygen is being used, the more apparent oxygen utilization you have. So it's kind of an approximation of productivity, or an approximation of the amount of consumption that's going on in a system, is one way of describing it. Okay, so again, so in this case, we have the closer it is to the surface, the lighter the color, and the darker the color, the closer it is to the bottom. So you have really low temperature, but temperature tends to be very stable toward the bottom. But as I said, we're only scratching the surface here. So if we're focusing on a 2-dimensional distribution model, we're missing a big part here. And this is another example, or this is another way of looking at it, where environments can change substantially with depth. So in this case, we have at 26 degrees west in the Atlantic, we have the depth so you have temperature on the Y and depth on the X, and you have at the equator, the Tropic of Cancer and the Arctic Circle. So at the equator, you go from really high temperature to really low temperature. At the Tropic of Cancer, you have a slightly lower temperature, and it goes to about the same temperature, and then you have a really small variation within the Arctic Circle. So you have all of these complicated dynamics that can change substantially with depth. And so depth is an important part of the diversity gradient when we're talking about these systems. So we have, and there are reasons that have been used to explain why this might be. So this is an example of a comparison of elasmobranch diversity, so sharks and rays, versus marine bony fish. So marine bony fish tend to decrease in diversity sort of on a negative exponential scale, whereas elasmobranch diversity tends to be a little lower at the surface, gets a little higher, and then goes back down. And there are a bunch of reasons that have been put forth as to why this might be, including energy and primary productivity. You get much more sun at the surface. That's where a lot of the photosynthesis is taking place. And so there are more things to eat up there, but also just generally favorable conditions. It's warmer, there's less pressure. It's easier to live in a lot of ways at the surface than it is at the bottom. So with all of this in mind, I'm now going to talk about the main thrust of this project in three parts. The first is modeling distributions in the open ocean. The second is a new perspective on marine biodiversity. So once we have those models, what do they show us? And then a little section on testing some of these older hypotheses with this new source of data. So now that we know where the biodiversity is concentrated, what is it telling us about what the potential drivers of that biodiversity are? So the first section, modeling distributions in the open ocean. So the first step, of course, is collecting data. And there is really good 3D, or there's really good environmental data in 3 dimensions for the open ocean that are provided by NOAA, the World Ocean Atlas. So this is the data that I used for the study today is from the 2018 data set. There was just a new release for the 2023 data set, so there's even more data available now. And what this is it's a depth-structured data set, so a lot of it comes from these instrument casts. So you drop an instrument and it takes measurements for temperature, salinity, these dissolved oxygen metrics at different depths. And then this is mapped globally. And so, as I said, this is a common source of this data, and it's often taken from marine cruises. However, it's really hard to get a ship like this up to take measurements closer and closer to the poles because of things like sea ice. So what happened in 2018 is they added some additional data sets from pinnipeds. So they actually glued CTD monitors to the heads of seals. And then they used the depth and latitude and longitude of these seals through satellite tracking, and they transmitted live temperature and salinity up near the poles. So that was a really cool source of additional data so that we can get some better polar information. But yeah, so the environmental data that I was looking at for this study today is apparent oxygen utilization, which we've just talked about, and temperature and salinity. So these are things that seem to really be things that drive and explain a lot of fish distributions. So the next thing I did was collecting occurrence data. So my plan was to get latitude, longitude, and depth for a whole bunch of different species in order to do this work. Depth being the key factor here because, as I said, a lot of these previous studies only use latitude and longitude. And the data sources for this were things like the Global Biodiversity Information Facility, GBIF, which is a global data set of both community science initiatives, but also digitized natural history museum records. OBIS, which is also a lot of natural history museum records. That's the Ocean Biodiversity Information System, as well as the literature and museum visits. And this is part of what I was going to do as a Marie Curie fellow as I got funding to do a lot of marine visits. However, my Marie Curie started in 2020. So with all of the spare time that I wasn't using going on museum visits, I instead took this idea of marine distribution modeling to a different level and focused on the computational problem of modeling distributions in 3 dimensions from my living room. So the problem was that a lot of these methods were developed for terrestrial systems. Although the algorithms will take 2D environmental data, latitude, longitude, that's it, that's all they want. But open ocean environments, as I've demonstrated, are better modeled in 3 dimensions. So I collected all those occurrence points, and then I went about solving this using R. And this is all now in a package called voluModel, which is available on CRAN if you want to use it. So I developed R tools to sample from 3D environmental environments and analyze and plot the results. So this is a basic niche modeling workflow with elements from the voluModel functions highlighted in blue. I didn't want to reinvent the wheel. I just wanted to make it easier to model things in 3 dimensions. So loading the environmental and occurrence data is facilitated through voluModel. You give it the World Ocean Atlas data set and your 3D occurrence points, latitude, longitude, and depth, and it will sample the environments for you, which you can then use to train your ecological niche modeling algorithm, whichever algorithm you want to use, whether it's GLMs or Maxent or any of your favorites. And then projections happen the same way projections happen for anything. And then I've also developed some tools for plotting and other analyses, because now that we have these 3-dimensional models, it's a little more challenging to visualize and implement using traditional methods. So the benefits, of course, are that we get more accurate models, and also this is an efficient and repeatable workflow for large numbers of species at once, because what I wanted to do was look at this broad-scale biodiversity pattern, so I needed to generate a lot of niche models. So as a worked example to show you a little bit how this works and why it might be useful, this is the luminous hake, this handsome devil up here. So that's a deep sea codfish species, which happens to be a little bit bioluminescent, hence the name. So here is a map of the occurrence points that I had for this species. The red dots are where they're found, and then the gray area is the study region that I used. And this is the distribution of depths that were available for the luminous hake. So 0 to 600 meters. So we had quite a few at around 400, and then again at around 100. So when I extract environmental data for the luminous hake from the surface, the bottom, and at depth, this is the distribution of environmental data I get, which demonstrates, again, why doing this in 2 dimensions might have been a problem. If I had done it at the surface, I get a really narrow picture of what the temperature and apparent oxygen utilization of the species is. If I did it at the bottom, it's much broader. But again, these species aren't necessarily, this particular species isn't always found at the bottom, it's found somewhere in the middle. And so we get a much different picture of what suitable temperature and apparent oxygen utilization is like if we actually draw it from where the species was sampled. So then, this is again a comparison of how the models look when I do this. So the surface models for luminous hake show suitable habitat all throughout this gray area. The bottom show suitable habitat mostly along the continental margin. So you get very different pictures, whether you're using the top or the bottom. But if you do the model in 3 dimensions with, and you can see that shown here, what you're seeing is the warmer the color is, the closer the suitable habitat is to the surface. The cooler the temperature is, the closer the suitable habitat is to the bottom. The more saturated it is, the broader the range of depths at which there is suitable habitat. So basically, this is being mapped as a series of transparent slices through depth. And so the more of those slices have suitable habitat, the more saturated the color is, like I said. So we have mostly really deep suitable habitat out here in the Atlantic, shallower, closer to the continental margin, but along the slope from the continental shelf down to the deep sea, you have a much broader range of suitable habitat. This red line is a transect that's shown here. So this is the same area shown in depth. So we have latitude on the X, and now this is depth on the Y. This hole right here is Cuba. So the red habitat, or the red area is the suitable habitat for this species. So we can see where the suitable habitat is positioned along this transect. So you can see not at the surface, not really at the bottom, but mostly in the middle, although it's shallower here than it is down here. Okay? So that's the first chunk, the takeaways being that these are new tools or new open source tools for 3D niche modeling, and that the results give a more accurate, depth-structured picture of species distributions. So once I had this worked out, the next step was to look at what it's going to tell us about open ocean biodiversity. Where is this biodiversity concentrated, and what can it tell us about the drivers of marine biodiversity? So the first step was to do the species distribution models. And again, as I mentioned, I focused on the Atlantic. This was mostly because the Atlantic is a very well-sampled area of the world for marine species. And it's a good place to concentrate my work at the beginning and work out all the kinks before then expanding out into the Indian and Pacific Oceans as well as the Arctic. So I focused on 3 fish families for this. Balaniformes, which are the half-beaks, flying fishes, things like that. These are mostly very epipelagic fishes. They mostly occur at the surface. Scombriformes, which are tunas and mackerels and allies that are mostly pelagic and very fast swimmers. And my favorite, the Gadiformes, the cods and allies. And these are mostly demersal and a little bit pelagic in nature. And the reason that I chose these 3 groups is I wanted to see if these different ecological utilization strategies might influence the differences in biodiversity patterns among fishes. So once I had this worked out, I then did, and I did my species distribution models, I then summed these distributions to map diversity. So I had a series of 3D layers, so I might have something like this, where you have latitude and longitude, and then depth at each of these slices. The blue might be the area where there's suitable habitat, and so then I summed all of these cubes together to take a look at the diversity patterns. But then there's another challenge, because once I have these things in 3 dimensions, how do I look at them? Because we often look at things in terms of 2-dimensional flat screens, and so if you had a distribution that looked like a cone, it ends up looking like a surface if you're looking at it, or as a circle if you're looking at it from above. So how do I figure out how to look at these things and understand this pattern? And this is where a conversation with my PI came in handy. So he is a big aquarium person, and he was like, well, what if we just modeled it to look like a fish tank? So we started calling these fish tank plots. So what you're seeing here, this is the diversity pattern. The higher the diversity is, the yellower it is, the lower the diversity, the darker purple. This red line is a transect across that you're seeing here. So again, like the one I introduced before, this is latitude on the X and water depth on the Y, and you're seeing the distribution of biodiversity here. And these colors are mapped directly to these, so these are directly comparable. But of course, this only shows you one particular transect, so what I then did is I animated it. So that's what you're going to see in the next series of slides. So keep in mind, this red line corresponds to the Y, this corresponds to longitude, and this corresponds to latitude, and then depth. So for our first set of results, this is Balaniformes, the flying fishes. Remember I said these are mostly epipelagic, so you see a lot of the diversity focused here at the surface. As the red line moves across, you see it tends to stay at the surface and is mostly concentrated fairly close to the equator. So these kind of show a very classical pattern of the latitudinal diversity gradient. Higher diversity, the closer you get to the equator. I'll let this run through again because there's a lot going on here. So again, you see the diversity is mostly here. This is the equator right here, and a little bit further out, but it mostly stays focused near the surface and at the equator. We'll just let this run through. Okay, so next up are the tunas. Remember, the tunas are more pelagic in nature. And you see that the diversity pattern is a little bit different. So now we get higher diversity here, but it's a broader span of diversity in terms of latitude. And you also get more diversity at depth than we did with the Balaniformes. So I'll let this run through again. So you see there's diversity here, mostly at the surface, but then it sort of moves out into the open ocean and drops down a little bit in depth. So it's not found right at the surface. It's sort of hovering underneath right here, but above 1,000. So that's where a lot of the primary productivity is focused. You don't really see a lot of tunas diving much deeper than that. So they tend to stay up here where the food is. The last one, Gadiformes. So this is, of course, again, the demersal group, and you see a lot of the diversity is now very differently focused. So we have high diversity at the equator, but you also have spikes in diversity up here in the temperate latitudes, and down here also in the temperate latitudes. So you have sort of this almost trimodal pattern of diversity. And again, we'll show this at depth, so you can see the diversity is kind of focused down here and sort of along the shelf a little bit. And then moving out into the open ocean, you have this more submerged pattern of diversity, and it's very spread out compared to the tunas. Let this run through again. Okay. Another way of looking at this, since I've sort of framed this in terms of latitudinal patterns and diversity, so if I did 2D models of these, which I did for comparison purposes with historical ways of doing this, the 2D models for the flying fishes show a fairly standard latitudinal diversity gradient. You get a broader diversity gradient with the tunas, and then sort of a more submerged with the gadiformes. But then if we do the 3D models, you can see at the surface you get different patterns than at 200 meters, which is sort of the limit for what's called the epipelagic zone. And then below 1,000, light doesn't penetrate in the ocean. So you see with the epipelagic group, most of the diversity is at the surface. For the tunas, it's more spread out across the surface down to 1,000. And for the codfishes, it's mostly 200 down to 1,000. So what we've seen is that 3D models give a novel picture of where diversity is concentrated, and that different fish families have different patterns. And so now the next question is why? So the last part that I'm going to talk about today is testing some of these older hypotheses using this new biodiversity mapped data. So what are potential explanatory variables that may be influencing these patterns? So again, the explanatory variables that I tested included latitude. Latitude on its own is probably not driving the diversity patterns, but there are other things correlated with latitude that may be explaining some of this. Also nitrate, which is a nutrient that can limit primary productivity in the open ocean. Also temperature, again, the warmer the temperature is, it's hypothesized that you then get more diversity for a variety of reasons, including primary productivity and even things like evolutionary rate. And then for the 3D models, I also did depth. I didn't do depth for the 2D models because that wouldn't make any sense. So what I did for each of these data sets, so I did 2D models and 3D models. I sampled 5,000 points per data set, so 5,000 points from the surface for the 2D model, or across the 2D models, and 5,000 points throughout all of the depths where there was biodiversity for the 3D models for each of the 3 families. So that's 6 different GLMs that I then did to to understand which of these explanatory variables correlate most with biodiversity. And I used a GLM with a negative binomial for those of you that are into the statistics. So this is the model fit and explained variance from these. Again, so I have each of the fish families split out, and then 2D models versus 3D models, and then the AIC score and the R squared. And what's interesting about this is that for the 3D models, they're the ones that have the AICC scores that are lower, which means that they're able to predict the amount of biodiversity better. But the amount of variance is explained more by the 2D models, which is an interesting perspective. So the 2D models might be giving us a strong but potentially spurious signal because there's less variance in these. Which makes sense, again, if you remember back to the histograms or the box plots that I showed you of what happens when we sample from the surface versus the bottom versus in 3 dimensions. So let's look at what happens with some of the hypothesis testing. So this is a table showing the correlation coefficients that I wanted to test. Any number that's shown in bold is statistically significant. And I'll sort of walk through these to make it a little more dynamic so you're not just staring at a table. So in the first case, latitude. So what's interesting is that the signs of the coefficient are opposite for 2D versus 3D models in terms of latitude. So it's a positive coefficient for 2D and a negative coefficient for 3D in all three cases. So this is saying that for the 2D models, latitude is positively correlated with diversity. So the higher the latitude, the higher the diversity. Whereas in the 3D models, the lower the latitude, the lower the diversity. So the closer you are to the equator, the higher the diversity. So, but these are such small numbers that you're really not seeing a whole lot of effect of latitude on diversity. Nitrate, the effect is a little bit higher, but again, it's still pretty low. But what's interesting here is that Bolaniformes have the highest correlation coefficient for nitrogen, so they're much higher than the other two. And it's positively correlated with their diversity. So the higher the nitrate, the higher the diversity of Bolaniformes. So more primary productivity in the epipelagic. This result kind of makes sense to me. Whereas for Gadiformes, you actually get a negative correlation coefficient with nitrate. So next up is temperature. And again, temperature is comparatively strong for Bolaniformes, but has opposite signs for Gadiformes. So warmer temperatures are higher biodiversity for the Gadiformes in 2D, but in 3D, it's actually the opposite. So the cooler the temperature, the more biodiversity, which is interesting. And then again, depth was not statistically significant for any of the families and wasn't relevant for 2D. So we don't really know what to do with that. That's not very conclusive. But as far as the takeaways from this section, 3D models predict biodiversity better, but explain less variance, which suggests and it also suggests that there are different processes at play for the three different groups of fishes. And then overall, the takeaways from all of these different pieces of the puzzle, I'm hoping that I've convinced you that the open ocean is not homogeneous and we can't just assume that what's happening at the surface or at the bottom is a good picture of what's happening throughout the entire water column, but also that efficient, repeatable big data methods are the key to understanding this marine biodiversity. So we've done it with fish. I'm hoping to do it with more fish and additional species. So the next steps are to model more species distributions and see how generalizable these patterns are. Also to investigate more explanatory variables. So as more data become available, we can take a look at other potential sources of data and also begin to explore how natural history may play a role in the biodiversity patterns that I'm seeing. So things like migratory pattern, reproductive mode, whether fishes are broadcast spawners versus live bearers, or other things could be potentially a big source of some of the biodiversity patterns that we're seeing. And also to begin to reconstruct the biogeographic history of these things in 3 dimensions. And of course, with a big data project like this, there are a lot of data sources that I'd like to thank. The Global Biodiversity Information Facility, the Ocean Biogeography Information System, the US, NOAA in the US, the section for biodiversity where I'm housed at the University of Copenhagen, as well as the National Institute of Aquatic Resources at DTU, which is also in Denmark, and then the Ichthyology Museum folks that I've talked to about this project throughout the years, including Randy Singer, Rob Robbins, and Peter Raskmola. And then for funding, a lot of this work was done first under a Marie Curie grant that again came about in 2020, but I'm continually supported by the Willem Center for Global Mountain Biodiversity and the Center for Macroecology, Evolution, and Climate at the Globe Institute at the University of Copenhagen. And with that, I will take any questions. Thanks. Yes?
Thank you for a really interesting talk. So when I think about global patterns of biodiversity, I think about tropical rainforests.
Mm-hmm.
And in the marine world, I think that's like the coral reefs or like the tropical rainforests of the ocean. What are their role in global biodiversity patterns and sort of what is their depth? You know, how does the depth, the third axis, play into, you know, coral reefs and these patterns?
Yep. Okay. So I'm going to rephrase for the folks at home. So you asked about the role of coral reefs in these biodiversity patterns and thinking about it in terms of like the coral reefs are the tropical forests of the ocean. Is that more or less? And how depth plays a role in these systems, and yeah. So in general, coral reefs are by far the most biodiverse area of the ocean, as you said, especially the coral triangle in the Pacific. So this is the area around the Philippines and Indonesia where everybody loves to go scuba diving. So what's interesting, so part of the thing that's interesting is coral reefs tend to be limited to areas where there's enough light for corals to for the zooxanthellae in the corals to photosynthesize. So most coral reefs are found above 200 meters. So that's why when you look at things in terms of the very most broad scale of biodiversity, most of the biodiversity in the open ocean is concentrated in that top, just because that's where the coral reefs are and that's where most of the species are. But I wanted to look at the open ocean because part of the problem is because all of the biodiversity is concentrated in coral reefs, that's where a lot of the work is being done. And so that's where we understand a lot of this. But a lot of the open ocean species are actually the ones that we're that's where a lot of our food is coming from. And what I'm interested in seeing is the biodiversity of a lot of these, or the role of biodiversity in some of these more fished organisms that tend to be a little bit more neglected from a biodiversity axis. Like, we're really interested in the productivity of cods, but we're not so interested in which species they are. And so that's kind of why I've been looking at that particular question. Does that answer your question more or less? Yeah. Yeah? Yep.
Super cool to see it. That should be very happy. So if I remember my phylogeny of fishes very well, which I may not, gadiforms are a lot older than the tubes and needlefishes, right? And this reflects an ignorance of evolutionary processes. But is there any role for like lineage age or evolutionary age in the diversity that you end up seeing particularly? So would you expect that because the gadiforms are older lineage that would lead to higher than expected diversity given all else? Does that make sense?
Yeah, so I'm going to rephrase again, and let me see if I got it correct. So you're asking about the role of phylogeny in this, and the idea that codfishes diverged much earlier than Scombriformes and Balaniformes, which is true, yeah, and then about the role of the age of that diversification, or age of that split in the biodiversity patterns that we're seeing. Yeah, so that's entirely possible, and especially because the cods did diverge so much earlier, and they're also deeper, which is interesting. So there's this other set of hypotheses that the deep sea is the oldest part of the ocean, and the idea that these codfishes are the older lineage within that, and they're inhabiting these older areas, and that's why we might see more biodiversity piled up in those areas. The problem is the deep sea also tends to be not a very friendly place, and so there tends to be a stronger extinction pressure in these particular systems. And so it may be that the fact that it's so unfriendly, may be limiting the amount of diversification, which makes it interesting compared to the idea that the tropics in a terrestrial system are the area that have the most diversity because it's the oldest. So that's another reason that working with these deep sea fishes is potentially really interesting, because we can potentially look at the role of age in a completely different way in a marine system compared to a terrestrial one. Thanks. I don't know, who do you see? What do you think?
My question.
Yeah, okay.
So how does this map, if you have maps of marine reserves, how does your new maps overlay with those?
So we had a question about how well these biodiversity maps overlap with marine reserves. So in general, I think a lot of the biodiversity patterns that I'm seeing, there tends to be a lot of diversity in the open ocean in areas that aren't protected. And part of that is that they're international waters, and so we'd need much more international cooperation for coming up with marine reserves in those areas, which I would suggest might be a good thing to try. But yeah, I haven't actually done the mapping exercise, but I think that would be really interesting to look at and sort of make that point that there is all this yes, there are all of these charismatic marine reefs where a lot of the protection is being focused, but in the open ocean and a lot of these areas that aren't being protected, there's actually a lot of productivity, and that's where a lot of our food is coming from. And having those marine reserves available to protect that productivity and to allow the fish populations to build up in these areas might be very beneficial. Thanks. Mm-hmm. Yep.
A really interesting talk. My question might be really naive. I know water is a really great thermal. This just doesn't apply, but I know in terrestrial ecosystems, things like seasonality and climate extremes also play a large role in structuring who appears where.
Mm-hmm.
And I was curious how you considered that here, or how that might be incorporated in examining explanations of the distributions of species.
Yeah. So the question is about seasonality and environmental extremes. So in general, it's definitely a bigger issue in temperate and Arctic habitats and the closer to the surface. So as you, in that three-dimensional drop-off I showed with temperature, it tends to drop off to the exact same temperature. So the deep ocean is mostly at 6 degrees centigrade, and it just stays there all year long. But you can have very big temperature swings, especially the closer you get to the poles, you get much bigger temperature swings. But I haven't incorporated any of that yet, but I would really like to look at temperature seasonality, especially with regard to these groups of fishes that tend to have a lot more of their diversity concentrated at the surface and in more temperate and polar areas. So this is actually where, getting back to codfishes, it becomes really interesting because a lot of the polar codfishes, the juveniles live in sea ice. That's where their nurseries are. So they're actually eating copepods that are eating algae that grow on sea ice at the surface. And then they they go to depth later. But yeah, a to-do list item. Yep.
So you mentioned early on that these geographical or ecological niche models were developed in terrestrial systems.
Mm-hmm.
There's been a long history of a conversation in those terrestrial systems about some limitations of these niche models that are based solely on abiotic conditions because they neglect biotic interaction. So for instance, a species might be able to occupy a physical environment but not occur there because a food resource doesn't exist or natural enemies do exist.
Yep.
And then the other conversation has been about disequilibrium. So potentially they could occur there but have not yet arrived because their geographic range is dynamic. Is that an issue for these marine systems? Is there a similar conversation going on in these types of fish systems in oceans?
Yep. So the question was, so in terrestrial systems, we talk a lot about limitations of biotic interactions, so things like available food resources, but also like competition sometimes comes up, and the role of disequilibrium in distribution. So that more or less, and then whether or not this is being talked about in a marine system. And distribution modeling is so little done in marine systems that I think we tend to we tend to ignore a lot of those caveats at the moment, just because there hasn't been enough distribution modeling done. Whereas in terrestrial systems, we've had the luxury of being able to do this for longer and we have more data. So I actually started distribution modeling in terrestrial systems and then sort of incorporated it back with my love of fishes later. It's definitely an issue, and especially for targeted fishery species, we may be looking at things that have been locally extirpated, and that's why they're not in a particular place. Sometimes we can approximate this by being really careful about our sampling, and we can potentially correct for it with bias surfaces and other statistical methods, but it's always going to be a caveat when we talk about this stuff. We need to emphasize that it's potential distribution, potentially suitable habitat, and not the actual distribution of the fishes, unless we incorporate some of these biodiverse or some of the biotic interactions and dispersal ability and things like that. Thanks.
which contrasts with my picture of tropical terrestrial regions, because they generally have really old soils and they're heavily leached, and so there's just less nutrient availability. And so something I've been thinking about is whether actually in tropical forests there's more diversity because there's no nutrients and species have diversified to try and find different strategies to get nutrients.
Mm-hmm.
So in marine systems, where do the nutrients come from? and why is it different from tropical terrestrials?
Yeah. So the question was where nutrients come from in these marine systems. And so there are a couple of different places they can come from. So environmental runoff is a big one, especially when we're talking about coastal communities. So the closer you are to a river, the more nutrients you're getting as runoff from the river. But what's really cool about marine systems is that you can also see a pattern of the effect of winds. So you have upwelling, especially if you look at the classic example is off the west coast of South America. You have the Guano Islands, and this is an area that's really, really, really productive. And it's not because of runoff, it's because of upwelling from the deep sea pushing all of these old nutrients up. And there's this really high productivity area along the tropics right there because of this upwelling. So it's not all coming from the surface, some of it's coming from the deep sea as well. So you have these really interesting combination of dynamics.
So how about functional diversity? Right?
Yeah.
So there's totally, and again, you got a real terrestrial audience here.
Yeah.
But it's like, so you have these species, and I guess Chris's question got at this a little bit with the phylogenetics, but
Yeah.
is there a way of looking at the functional diversity of fishes, and does that show the same pattern as the total diversity?
Yeah, so the question was whether the same pattern, we're seeing the same patterns with functional diversity as with just regular alpha diversity, species richness. And I haven't looked at this directly, but if you look at some preliminary work that's been done on functional diversity, at least as far as like gross geometric morphometrics, like the shapes of fishes and how they change, the diversity is mostly found in the deep sea. So you see all these wild things with huge mouths that are super long, or maybe they have, you have all of these angler fishes with these crazy escas that are being used to lure in fishes. And so you're actually potentially seeing a whole lot more functional diversity in the deep sea than you are in at the surface, where everything is just kind of fish-shaped because everything has to swim really fast to grab stuff, whereas in the deep sea there's a whole bunch of different strategies that you have for this.
Is that because of the unfriendliness of the deep sea, or because of the constant conditions?
Yeah, so the question was whether it's the unfriendliness of the deep sea or the constant conditions, and I think it's some combination thereof. So everything's the same all the time, and so there's maybe less pressure to conform to a certain optimum, but also there is so little food that there are just so many different strategies for how to catch that food, and so few mates that there are all of these like crazy ways of attracting mates, either with chemosensory signals or with light signals, and you have all of these like secret light signals that are only at certain light frequencies that only certain fish are adapted to find so that you can avoid predators while still calling in mates from somewhere else. It's wild. I love it. I love the deep sea.
I just wanted to take a moment. You can ask Chris a second. I wanted to give a second to ask some questions from online.
Yeah.
We had a question from someone who said, you mentioned citizen science in passing. I'll just note here that we often use community science instead, so we can include non-citizens. How is this done and who is involved? So how does that work for marine systems, having community science data?
Yeah, so in marine systems, there's not as well. So a really great community science initiative that's available is we can pull in recreational divers to run transects while they're doing recreational dives. So we ask them, they get trained in fish recognition, and they say you give them a board and a wax pencil, and they count how many fish they see along a particular transect, or they report the fish they see. I myself have contributed to community science by going on iNaturalist and reporting a splendid toadfish I saw off of Cozumel, because who wouldn't want to report a splendid toadfish? They're amazing. But yeah, so it works the same as it would in a terrestrial system. iNaturalist works very well. Sometimes we get things like fishing rallies. So if we can get reports from like sword fishing derbies and things like that, potentially as well, depending on how willing the fishermen are to report where they found their catch.
Another question we had from, another question we had from online, there's someone who asked, will marine plants be included at some point in the future?
Marine plants. I would love for marine plants to be included. I've been talking with some folks on doing this with phytoplankton especially, because we have some really interesting depth dynamics with phytoplankton and turnover. But it's still in the early stages. We need to get funding first. Yeah.
I guess I'm trying to think of how to phrase this. As you get deeper, do your data that you're basing the models off get less reliable, or are diversity patterns with depth related to the abundance of the particular species and family you're looking at? Mm-hmm. Do you get less certain that you would capture them?
Yeah. Okay, so the question was whether as we get deeper, we're less certain about the data that we get. And that's certainly true. And part of that is just the way these data are, the way that the deep is sampled. So a lot of times it's coming up through pelagic trawls. So they open the net at one depth and then they drag it up to another depth. And so you get this really big range of possible depths at which a fish was collected. So that needs to go into the model as well. I didn't report this particular aspect, but I'm working on this a little bit in terms of marine, with museum data specifically, and the degree to which sampling effort may be structuring some of our understanding of biodiversity. And it does look like it's playing a role. So part of it's just going to be trying to get more funding to do more detailed depth sampling using things like mock nest nets, which are have much more precise opening and closing depths to get better, more precise data for depth.
Are those samples generally non-destructive?
They're very destructive. Generally, when these kinds of collections are being done, they're being done specifically to put things into museums or to eat them. A lot of times, especially when you're doing deep sea sampling, if you pull things up, they're going to be pretty beat up by the time they get to the surface. So they're generally not being thrown back. For something like longline fishing, that's possible. So sometimes things are sampled just by, old-fashioned rod and reel and things like that, and those can sometimes be non-destructive. And but you have to be really careful about how quickly you reel them in, because they can go through the bends just like we go through the bends. But usually it's the swim bladder comes out of the mouth. So. Okay.
Well, we can all go to lunch now if we're done with having questions. Lunch is good too.
Yeah, lunch is great. Thank you all very much for your questions.